Emil is one of our accelerator finalists working on Cryonics. We have JJ, a fellow here, and Stephen, a fellow as well, and then two fantastic newcomers to the Foresight community. So I'm very, very, very fortunate to have you guys on board here. Thank you so, so much for coming. Okay, we're starting with the first slide that I can get access to here. Who do we have? It was a little bit like Russian roulette here. Oh, there you went. Emil, Kendziorra, you're up next. Tell us a little bit what it is that you're working on, what is a long-term goal of this field, and how can people solve that challenge? We already had a cryo discussion during the lunch break, so you can pick up from there.
Thank you so much. So basically what we do, right, is if the guys from the panel before me don't make it work, don't make anti-aging, longevity, or life extension work within the next, well, let's say 10, 20, 30, 40, or 50 years, which of course I unfortunately don't believe, and I very much hope I'm wrong with that, right? I very much hope that I need to do the apology tour in 20 or 30 years and need to tell everybody that I was always the pessimist and the other guys were wrong, or right. So I very much hope that will be the case. But of course, I wouldn't do what I'm doing if I wouldn't believe that, or if I would believe that, right? Because in fact, I actually come from the longevity space. So I studied medicine in the beginning, did cancer research and so on, to always wanting to, due to the reason of wanting to go into the longevity space. And then I did a bit of a detour with building a few tech companies, but now I'm kind of back at the same value proposition, of course, but taking a different approach. And of course, most of you might be aware of that approach, right? This idea of cryopreservation, right? And the fundamental idea here, of course, is that for people who would like to live significantly longer than currently possible, while being healthy, of course, it's always a combination, of course, of living longer while being healthy. But let's assume this will take significantly longer than sometimes optimistically being suggested. Then there might be a time when people still need to go into, at their end of their life, or when they're being diagnosed with any terminal disease, might be cryopreserved for a while. Until then, such technology to make them live longer or enable them to live longer exists. And this is what I'm building. Of course, this is not a new topic, right? This has been around for a long time, so this is not something fundamentally new. But what I think what is fundamentally new to approach this topic slightly more in a slightly more professional maybe way, and maybe in a slightly more accessible way for a larger audience. So we're building, or I founded two organizations in Europe. Currently mostly active in Europe. One is called Tomorrow Biostasis, based in Berlin in Germany, which does day-to-day cryopreservation. It's the operative company, right? If you want to sign up with a cryopreservation provider, you can do so. We staff the medical teams, we hire the doctors, and so on. And then, of course, everybody's well aware that, well, while you might be able to cryopreserve people right now, you cannot take them out of cryopreservation yet. And of course, this is a big issue. And then the second, it's the most important issue potentially, right? So we're working on that. And that's the second organization that I founded. It's a non-profit research foundation and research institute that will, in the next couple of weeks, open a research institute that we've just built close to Zurich in Switzerland that will do research just for this topic.
Okay, give us a long view too.
Yeah, so the long view, of course. I mean, I don't know actually what I put there. I would say biostasis and cryopreservation is the tangible path of longevity, right? So at some point, I'm very positive that at some point longevity technology will be around. The only contention I have is when that will be, right? And of course, for everybody who is being diagnosed with terminal diseases right now, next week, next year, whatever, in 10 years, you need a bridging technology, and this is what we're doing.
Good. Wow. That was an abrupt ending. JJ, it's your turn. Yes.
Jay.
Yes. Okay, great. We have a challenge here from Emil as well. And we're moving over to JJ. And now we're really getting into the bio-risk AI territory. I like it.
Yeah, okay. So I'm JJ. I work for InQTel, a strategic venture capital firm that does deals on behalf of national security. So I'll be talking about biotechnology and artificial intelligence. And one thing I would like you to get out of this is this idea that it's very promising. There's a lot of promise in these two words together. And the other thing I would want you to get out of this is how to make it promising right now, because right now it's still very primitive. The general idea is, you're all familiar with how in deep learning and the recent advancements. It's not that recent at this point. It was recent at one point, but in computer vision and so forth has led to models that are better than human performance on various tasks like identifying objects and images and so forth. Those tasks are really hard. They're not easy. A picture of a cat, maybe to you and me, is obviously a cat, right? But what makes a cat is an image of a cat is a very complicated thing. It's like there's different poses and lighting conditions and different kinds of cats, and so how does a model know that so well? And the reason is quite simple. It's given a lot of data, right? So the advancements in the field came from really two things, more than anything else, is the quantity of data, the data set ImageNet, and also the computational advancements using GPUs, which is why NVIDIA is worth so much these days. The models themselves also advanced, but they were advanced kind of incrementally. Convolutional neural nets have existed since the 80s, but have been used in these kinds of tasks since the 90s. So the problem in biology is it's also very hard, but it's not really significantly harder. So it's a similar hardness to computer vision. So why is it not as advanced? It's because the data is just not there. So that's kind of where I want to go, is create more data in the field.
Anything in particular?
Yeah. So I have the key challenges here. One of them is called life on Earth. So very easy to describe. You just sequence life on Earth. So sequence as much as you can of different species and stuff, and their phenotypic and omic information. You tie them together in a really nice, easy-to-use data set, and you make it public. You make it really big and not have the kinds of problems that typically exist with biological data. And then the other is chemical property prediction. So example is we have data sets, but there are thousands of toxicity of small molecules. What if we, instead of 8,000 examples, we had 80,000? We'd probably make more progress, right? So more kind of advancements there. And we could get there with stuff like organ on a chip, which is a technology for doing kinds of assays, even on human cells. So we get even better data that way. And that's it.
Wow, lovely. Okay, you were actually quite concrete there. Okay, even though it's a large challenge, obviously. Okay, JJ, thank you so much. Next one up, we have a new slide edition here, which just came in. Let's see if it loaded. And ta-da-da-da-da. Wow, we're going places with this slide presentation. I love it. Okay, here we go. How do you like my collage? Okay, Ashut, what are you working on? What's the very, very long term of this, and what's the challenge that other fields can solve?
Sure, yeah. So I work for Deep Genomics, which is a machine learning for drug discovery company, and what we're trying to build is programmable medicines. So that means that if you want to make a specific biological intervention, we want to be able to engineer those. You give that to the machine learning system, it gives you a compound that does exactly what you want. And what has enabled that is, one, the machine learning AI revolution that JJ was talking about. But I would actually really disagree with the idea that it's only a little bit more challenging. And the reason why is that for image recognition, we're actually aiming for maybe human level performance. But in biology, that's not the case. What we want is superhuman level of performance, orders of magnitude better predictions than humans can possibly do. And I think that is a more challenging thing than just copying human level performance. But the other enabling technologies are very large scale biological data sets. The last panel we talked about that. But I think one thing that is maybe underrepresented in that is the huge amounts of human sequencing data that we're generating. And why I think that's so valuable is that it gives us hundreds of thousands and millions of experiments on actual human beings that have lived an entire lifespan. Which genetic perturbations lead to better health is something that we can extract from that data and identify what genes and proteins we want to change in order to prevent and treat disease. What was the last one? Oh, right. And then the other thing is nucleic acid therapeutics. And I think that's really key to building programmable medicines. And that's because when you look in our bodies, how do our bodies programmably change gene expression? And the answer is nucleic acids, whether it's transcript levels or microRNAs, or in the case of bacteria, the whole CRISPR system. It's all nucleic acids that are driving the programmable nature of protein production. By combining all three, we can produce programmable medicines and then treat all genetic diseases. And I want to focus on the idea of a genetic disease. Some people think about it in a narrow way. They think about monogenic disorders. So diseases caused by you are missing a specific protein, and with 100% probability, you get a disease. But I think that's way too narrow. I think that almost everything is a genetic disease. And the reason why is that when you look at heritability of many, many phenotypes that we care about, they're all heritable. And that means that they're all genetically, maybe based is the wrong word, but can be changed genetically. And so I think that almost every disease is actually a genetic disease. And so if you can produce these targeted interventions, you can treat or prevent almost every disease. And that's what we're working on at Deep Genomics. In terms of what challenge do I want solved, I want to be clear, I don't want to solve this. I want someone else to solve this for me. And that is...
That's what you get to do here. You
Yes.
just put something really hard out to people.
And that is ligands to enable cell type specific delivery. In the nucleic acid field, there's a conjugate called GalNAc that when you add it to any nucleic acid, causes it to be taken up very productively and specifically in liver hepatocytes. And this has totally revolutionized how we can treat a wide range of diseases that are caused by proteins in the liver. And what I'd like to see other people do is solve that for every other tissue. So that way we can achieve delivery to any cell type we want in a specific way that crosses the various barriers that get in the way of reaching the CNS or reaching, there's actually apparently an analogous to the blood-brain barrier in the testes that prevents therapeutics from getting there. And so if people can solve those challenges, that would make my life a lot easier and I think really accelerate how we can use nucleic acid therapeutics.
Anyone have an idea? Okay, you get to think a little bit. That was a quick hand. Okay, good. Very nice. Okay, that's also a concrete challenge here. Next one up, we have Steven. Steven, what are you working on as a Foresight Fellow? You just recently, Steven, gave a fantastic presentation to our Biotech and Health Extension group and got lots of emails afterwards asking for your email. So here we go, Steven.
Great. Well, I love cold emails, so that sounds great. I mean that, I'm not joking. Yeah, so I actually feel like I've had a lot of my work done for me here because people keep talking about gene therapy and doing a better job than I would of explaining why it's exciting and what it is. So what I do is I work at a company called Dyno where we're trying to engineer a specific class of gene therapy vectors, which is AAV, a Dino-associated virus. It's a very commonly used viral vector for current gene therapy clinical trials, and also a bunch of like experimental studies in mice and other animals. And we're trying to engineer that vector to be much better for gene therapy in a number of ways. So we work on engineering these vectors to be better at targeting certain tissues and also better at detargeting other tissues. We're working on trying to make these vectors less immunogenic, and a variety of other things. And our approach to this is very machine learning based. So we're trying to use the combination of like good biological techniques that have come around, like high throughput sequencing and synthesis are the simple ones, and also some of the things that enable this sort of multiplex in vivo screening that Martin did a good job of describing, to characterize the fitness landscape of AAV for these various things we're trying to target, and then create optimized AAV vectors for various diseases, tissue combinations. You could think of it as like customized AAV vectors for whatever gene therapy application you're trying to work on, so that people like Gordian or some of the people working on reprogramming or more traditional descriptions of genetic diseases, which is actually what we're focused on now, can be solved. Because right now there are just a bunch of challenges that we believe revolve around current AAVs just not being good enough as delivery vectors to consistently have like high success rate clinical trials, especially in humans. And so I think if we can solve this problem of like having really safe, effective, targeted delivery, gene therapy provides this really flexible toolkit for solving lots of different types of diseases, and an especially nice toolkit for attacking problems in like longevity.
Lovely.
Well, my challenge is actually similar to what Amit said, not something I necessarily want to solve, but it is something that, as I've become more familiar with the industry, I think is a really missing thing, which is there's a lot of talk about how like mice don't necessarily transfer well to humans, and there's a lot of good work done on like humanized mouse models. In gene therapy, a lot of people work with non-human primates because they're a better model, or they're thought to be a much better model for humans. But at least I haven't seen a lot of work on like characterizing relationships between these model organisms and humans for like specific interventions. And like, I'm a machine learning person, so I think about this from a machine learning context, but like, it doesn't necessarily have to be machine learning. Like, I'm just interested in people doing more work to try and actually like understand the mechanisms behind when things transfer and when they don't, and potentially have data sets that actually can help us characterize this. Obviously it's hard because getting human data is harder than getting model organism data, but I think this is something I've been thinking about and I'm excited about, so I threw it up there so that I am hopefully I'm not as galaxy brained as some of the other talks, but I'm trying to be a little more galaxy brained with this one.
Oh, lovely. Okay.
What's your view on like FDA hitting for AAV?
Wait.
Sorry. What's your view on like the FDA recent updates on AAV, and how are you thinking about that?
Which recent updates?
So I remember they were doing some investigation about like AAV therapy and
Yeah, I guess my not personal take is that obviously we are really excited about trying to deal with some of the problems that I think current AAVs, to a debatable degree, but like to some degree have when you put them in humans. So simple example is like when you dose AAV therapies in humans now, you're often doing like really high dosages, like you're giving just really large dosages of vectors containing your therapeutic payload in them. And some people believe this is partly related to why you've seen sort of adverse events in like the liver and things like that. And so we're very excited about ways to try and potentially reduce the dosage you need and also detarget the liver and things like that, so that hopefully some of these adverse effects become less likely. Does that answer your question?
yeah, fascinating.
Dina.
All right. Well, Allison, thank you for having me. It's my first Foresight weekend. I've had a great time, so thank you for inviting me. Just heads up, I've had a lot of coffee during lunch break and then rush to the interesting talks, and coffee is a potent diuretic. So if I start talking too fast, like just let me know. I'm a medical doctor by background, and first started working in longevity by building a longevity research clinic with Eric Verdin of the Buck, where I still have a post. I'm doing a lot of longevity investing through the SALT platform, and have recently started an early stage biotech in cell therapies for ovarian aging. So out of kind of working in longevity as a doctor, scientist, slightly an investor hat, and an entrepreneur, the area that I personally believe is probably the one that can be the most impactful at this stage is cell therapy and rejuvenation. Basically, just being very simple. If something is missing, replace it. So if diseases and aging occur because of organ and tissue damage, if we could engineer cells and replace them, that could potentially be curative. So I kind of agree with a lot that Jean was saying earlier. And I decided to focus on cell therapies as the most potent technology. So a couple of like enabling technologies that I'd like to see in cell therapies is number one, like a very good way of making purely allogeneic, non-immunogenic cell therapies. So I'm sure everyone here, because this is like a very selected crowd that is interested in biotech, has seen Vertex data for their allogeneic cells for diabetes. And even in that trial where they reported curative results, they still immunosuppress patients. So I'd like to see completely allogeneic cells that can be used for same cell line for all of us here without the need for immunosuppressive therapy. We could be there. We have like ways how to make HLA knockout cells, but we haven't done it fully clinically yet. The second thing is mass production of cells. So anyone here who has done work in iPSCs even knows that it's very difficult to produce iPSCs. With just Yamanaka factors, like 2% yield. So if we wanted to supply cell therapies for all of us, we need very good manufacturing facilities. And that is something that I'm seeing in a lot of new technologies. We're, for example, seeing it in the food space as well, just like in medicine, is that it's very difficult for new technologies to scale because there's just no manufacturing facilities built for these technologies because previously there wasn't a need. So manufacturing and ensuring that it's an efficient process with same batch potency. And then the third one truly is like increased patient demand. So if more people were kind of demanding and just understanding that health is not something that we should compromise with, that aging or rejuvenation is just not something that we should have to accept as a fact of life, that would just increase kind of a bigger demand, which would push the players like pharma and everybody else to create the supply. And I think we do need to have we can't just work in silos in this aging space, and we have to find a narrative how to change the public perception of what they think about healthy longevity. So kind of the capabilities of this, I believe that reproductive longevity, particularly the field of IVF, is one of the first ways where we can see cell therapies in the clinic, which is why I've decided to work on it, so I'm a bit biased. The second thing is I think they can help us cure diseases that occur because of isolated organ failure. So for example, end-stage renal disease or type 1 diabetes that we're seeing with Foresight or Vertex. And then thirdly, which is kind of the biggest goal, and maybe we have to get cryopreserved before that happens, who knows, is to prevent age-related immune decline. And truly the end goal of this is to compress morbidity, which will hopefully also, I think, increase everyone's life expectancy. So for me, the challenges, I mean, there are companies like BitBio that's working with resilience. There's Seleno Bio, so there are companies already working on this. But if anyone has solutions around GMP certified, low-cost manufacturing of cell therapies, and like way to automate that, please let me know. I would love to chat, and hopefully there are smarter people who can help me solve that.
Great, I love that you actually all put a challenge out there. We're like, I don't want to solve this, but please, you guys go along with it. Any comments, any questions already from the panel to each other? Any one of you guys, question, comment already? Mark already, great. You're positioned very conveniently over there.
This is more of a reaction than a question, but feel free to respond. I don't like the positioning of the case for cryonics being premised on pessimism about how long longevity technology is going to take. There's a saying among cryonicists that you probably already heard that getting frozen is the second worst thing that can happen to you. I got my cryonics contract in 87. I did not expect longevity technology to take as long as it's taking. I expected not to need it. But great to have just in case. And I've had friends who've died because they thought they had plenty of time and they had an undiagnosed fatal condition. So I just want to really distinguish, separate the case for cryonics from pessimism about longevity technology.
Quickly responding to that. So fundamentally, I agree with you. So this is not something, this is not an either or, right? So the only problem where I don't agree with it, so it's basically a research allocation problem, right? There's only so much money for the general value proposition, I would like to live longer than currently possible. If everybody believes, just taking this pill, figuratively speaking, right, or taking this treatment is 10 years down the line, then there's no funding for cryopreservation. And we cannot overestimate how good cryopreservation already is. It's not a field where I would argue the quality of preservation is great so far, right? So the field needs significantly more funding, and if everybody thinks, hey, I just need to take this pill 10 years down the line, then there is no interest in that, right?
I don't want to take the time to do a debate, but I want to say this question that you just raised, I think, is very much worth debating. I think that, in fact, with regard to what we should expect of future technology, the quality of cryopreservation is fantastic and has been fantastic for decades. I'll just mention one phrase from Ralph Merkle that completely changed my perspective on this. Revival is cryptanalysis. And you look at how little evidence you need to successfully cryptanalyze, and then you ask, is there enough evidence in a frozen brain to cryptanalyze it into the most probable healthy brain that would have been frozen into this damaged frozen brain?
I hope you're right, but I'd much rather put more work into err on the side of caution in this regard, right? But I very much hope you're right. I don't think you are.
All right, up for debate, and we are conveniently breaking for the next. Well, we're meeting here again at 3 p.m. So I want to thank our panelists very, very much for coming on. This was a fantastic session that really went all the way out there.